Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic terms. And our motto is 5 terms at a time, keep the learning in the mind. The first term of today is codominance. Codominance, as it relates to genetics, refers to a type of inheritance in which 2 versions, alleles, of the same gene are expressed separately to yield different traits in an individual. That is, instead of one trait being dominant over the other, both traits appear, such as in a plant or animal that has more than one pigment color. In people, one codominant trait that you can't really observe by looking at a person, but many people know about themselves, is blood type. People with the AB blood type have one A allele and one B allele. Because both alleles are expressed at the same time, their blood type is AB. The second most common term is codon. A codon is a DNA or RNA sequence of 3 nucleotides that is a trinucleotide that forms a unit of genomic information encoding a particular amino acid or signaling the termination of protein synthesis or stop signals. There are 64 different codons, 61 specify amino acids and 3 are used as stop signals. When people talk about the genetic code, they are referring to the phenomenon of DNA containing an informational code to create the proteins that an organism needs to function. The DNA serves as a pattern for making messenger RNA, and the messenger RNA then serves as a pattern for making a specific protein. DNA and the corresponding messenger RNA are made up of a series of bases. In RNA, these bases are often labeled with the letters A, U, C, and G. A set of 3 bases makes up a codon. The messenger RNA contains many such codons, with a series of 3 bases followed by 3 more bases, and then 3 more bases, and so on. Each codon instructs the cell to start the creation of a protein chain, to add a specific amino acid to the growing protein chain, or to stop creation of the protein chain. For instance, a messenger RNA codon, GCA, signals the addition of the amino acid alanine to the protein chain. The messenger RNA stop codon, UEG, signals the end of that protein's production. The third popularly known term is complex disease. A complex disease, or condition, when discussed in the context of genetics, reflects a disorder that results from the contributions of multiple genomic variants and genes in conjunction with significant influences of the physical and social environment. For this reason, complex diseases are also called multifactorial diseases. This stands in contrast to a simple, genetic disease that is more directly caused by mutations in a single gene. Common examples of complex genetic diseases include heart disease, diabetes, and cancer. Complex disease really just means that it isn't caused by one specific gene. Many genes and variants contribute to it. Even your environment contributes to it. Often these individual contributions are so small that they're hard for scientists to really disentangle. Calling these kinds of diseases complex implies that other diseases are just simple, which I bet that a lot of people who work on simple diseases would disagree with. No gene or protein works in a vacuum, so no genetic disease is ever truly simple. The fourth commonly known term is congenital. Congenital refers to a condition or trait that exists at birth. Congenital conditions or traits may be hereditary or result from an action or exposure occurring during pregnancy or at birth, or they may be due to a combination of these factors. Congenital isn't really a great descriptive term for what it means. It's really just the traditional way that doctors communicate that a disease or disorder or trait was seen or at least thought to have its origins in and around the time of pregnancy and birth. So, terms like congenital heart disease, for example, refer to structural defects in the heart that are noted shortly after birth or at the time of birth and differentiated from more complex disorders like coronary heart disease, which usually develops as people get older. The last, that is the 5th term for today, is contig. A contig, as related to genomic studies, derived from the word contiguous, is a set of DNA segments or sequences that overlap in a way that provides a contiguous representation of a genomic region. For example, a clone contig provides a physical map of a set of cloned segments of DNA across a genomic region, while a sequence contig provides the actual DNA sequence of a genomic region. A genome is typically studied by breaking down its DNA into pieces, analyzing those pieces in the laboratory, and then computationally putting those pieces back together in a conceptual way. Years ago, before genome sequencing became routine, The pieces of DNA would first be cloned and then individual clones would be analyzed to determine which clones overlapped which other clones. A set of overlapping clones that together contain a contiguous region of a genome is called a contig, or in this case a clone contig. Genome sequencing uses the concept of a contig, but in a different way. For genome sequencing, the pieces are not actually physical segments of DNA but rather are stretches of generated DNA sequence often called a sequence read. By establishing the overlaps among a set of sequence reads, one can assemble a sequence contig, from which the actual sequence of the genomic region can be deduced. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.